Okay guys, we're going to launch into the specific side of our body defenses, the adaptive or acquired immunity. I always think in A&P that the immune system's the coolest, but if you think about in micro how we've learned that generation times for bacteria can be 20 minutes, right? For E. coli or staph is 20 minutes, 25 minutes. The fact that viruses can be replicated in much faster time than that means that they're going to evolve that much faster than we do, right? And yet we have an immune system that works pretty darn well against bacteria and viruses that cropped up throughout our lifetime. So our adaptive immune system's pretty cool, all right? In order to understand the adaptive immune system, we need to understand antigens. Remember that this side, the specific immune system, it was capable of recognizing something specific about pathogens. And that specific thing is an antigen, all right? If we think of the word antigen, it kind of makes sense because it's our ability to generate an opposition to these molecules, all right, that will allow us to protect ourselves. So antigens will generate that opposition, okay? Now, our immune system has to be able to differentiate between antigens. Oftentimes they are proteins or proteins with something attached to them that happen to be in our own body versus those molecules that happen to be on foreign invaders, right? Like bacteria or viruses or something like that. So our immune system has to be able to differentiate between antigens that are self or normal molecules, right? Versus foreign or non-self molecules. And the coolest thing about the adaptive immune system is that it has a memory. There's a memory built in here where once you're exposed, the immune system is going to be that much better and more sophisticated going after that very same antigen again later. Now, when we look at this adaptive side or the specific side, we generally break it down into two camps, two basic categories. One is humoral, and here we'd be talking about the humors of the body, the liquids of the body, and antibodies are found in those liquids. All right, so humoral, you'd want to immediately connect the word humoral with adaptive or specific, right? And also with antibodies, because humoral is automatically talking about those molecules that are dissolved in those liquids. So it has to do with the body fluids here. Cellular, the cellular side or the cell-mediated side of adaptive immunity has to do more with specifically the T cells, that type of lymphocyte that happens to be in our system, all right? So what we're going to turn our attention to soon will be that humoral and then our cellular side. But let's first get a good handle on these antigens. What are antigens? All right, antigens are substances that can activate the immunity. Like I said, right? They're capable of causing us to generate an opposition, okay? The degree to which we generate an opposition to an antigen, some substance with a specific shape, all right, is called immunogenicity. The degree to which an antigen, AG for antigen, okay, can cause us to respond with some kind of immune function. In this case, it may be antibodies, right? So the degree to which an antigen stimulates antibodies to be produced against it is immunogenicity. Now, not all antigens have the same immunogenicity. They all have different shapes, all right? And some people maybe respond vehemently, very much so, to a specific antigen. Their immunogenicity is high for that antigen, and some may not. We could also say that any one antigen will have many shapes associated with it. Something we call antigenic determinants or epitopes, specific parts of the antigen that would be recognized by the immune system. All right? So we take a look at this picture here, right? So this in red here could be an antigen. It could be a molecule that's sitting on the surface of a bacterium. It doesn't have to be, though. It could be a molecule that is secreted by a bacterium, like a toxin. So there's nothing saying an antigen, in this case I'm talking about foreign antigens, right? There's nothing saying a foreign antigen has to be attached to a cell or attached to a virus. It could be free-floating, something that is secreted by a bacterium, all right? But you notice here that this red molecule, basically it's a circle, right? But then you see these little shapes that are sticking off out either side, right? So here I have a kind of a triangular shape. Here I have kind of a circular shape. Here I have kind of a block shape that's sticking out. Each one of those would be an antigenic determinant. And antibodies, like you see in blue here, okay, these antibodies would have the ability to attach to any one of those antigenic determinants. All right? This is to say that a single antigen on a virus or a bacterium or a toxin or whatever, you could make several antibodies against that individual antigen because it's got all these little shapes, little mini shapes associated with it. Okay? I think of this, if we take this piece of paper here, okay? I'm done with this piece of paper. So if we take a look at this, this is the antigen, right? So if I were to look at this crumbled up piece of paper, if I throw it across the room and it's in the corner somewhere, it looks kind of like a wadded up circle, right? But if I pick it up and look at it closely, I've got all sorts of little mini shapes. Some kind of dive down deep, some are kind of rounded out on the outside. You could make an antibody against any one of those shapes, okay? Any one of those. And the more times you're exposed to that antigen, the more chances you might make more antibodies for those little mini shapes, those little antigenic determinants. All right, so antigens could be foreign or self, right? We could have antigens on our surface that we should not respond to, and these would be called self-antigens. An example of a self-antigen that is a really common one, and it's going to be a useful one later in this unit, so we need to know about it, are the major histocompatibility complexes, MHC, thankfully, right? So it's a mouthful. So we just say MHC proteins, and there are a couple different kinds of MHC proteins that are self-antigens on our cells. Some of these are found on all body cells, every cell that I want to look at, and some are just found on immune cells like white blood cells. So we're going to leave it at that here for the antigens. What we're going to do next time is we're going to talk about how these antigens might trigger antibodies to be produced against them. We're going to dive into that humoral immunity next. Okay?